Hello, everyone. My name's Andy Moreo. I'm excited to talk to you today about our approach to developing a lentiviral platform. I wanted to start with a brief introduction of Andelyn, and if you've heard anybody talk about Andelyn or any of us talk about our company, we tend to always talk about our name for good reason because that's what grounds us. It's what links us to where we came from. It's our namesakes are Andrew and Evelyn. Andrew was at the age of 8 was the first to receive the first U.S. investigational gene therapy drug for DMD that was a precursor to Elevidys. And then Evelyn, at 8 weeks old, received um, a experimental gene therapy for SMA that eventually became Zolgensma. So, that speaks a lot to us. It's what motivates us. It's what grounds us. So I have to talk about it every time. Um, I also like to talk about our timeline. Not to date myself, but I've lived through most of this. We've been at this for a very long time, starting with NCH and our very first work with AV in 1992, all the way to 2006, 2007, when we built our first GMP facility, which is when I joined, and developed at that time, we didn't call it Curator, but our Adherent AV platform. And then through our spin out in 2020 as Andelyn Biosciences, really to carry on that original mission, and continue to grow with the field that we had been working in so long. That we created a suspension AV platform, in dubbed it Curator, in all the way up through today, uh, where I'm really excited to talk about our lentivirus platform that we've also built on the Curator, with the Curator bones, so to speak. Um, so, who are we today? So, we still carry that original mission, that core function that started, back in the early 2000s as a core facility where we do work with academic institutes, but now we're a full service concept commercial gene therapy CDMO. And again, over those 20 plus years of continuous gene therapy manufacturing, we've manufactured over 500 batches, supported over 50, 85 INDs, and there's pluses next to all of these things, and we have 70 plus active programs that include PPQs at this point, going into commercial batches. So, you know, um, about the Curator platform, we wanted to build this lentivirus platform with the same bones for good reason because um, platforms bring a lot of good things to the table, and there's a lot of considerations, but there's a lot of considerations that go into program development and manufacturing strategies for any given drug product, all the way from program factors, with uh, program or indication, route of delivery, disease penetrance, the dosing per patient. Uh, and then there's what that platform brings to the table. It allows for scale down modeling to enable your process characterization, your PPQ work. It also brings standardization of cost of goods, standardization of assays. All of these things added together need to equal different quality attributes that suit an individual indication, whether that be one that's a traditional AAV gene therapy or a cell-based therapy that's using a lentivirus. Um, and I always liken it to an airplane wing, right, where its strength is in its flexibility. If an airplane wing is completely rigid, it's going to break. Um, Same thing with a platform. If a platform's completely rigid, if it's one size fits all, it's going to suit a very narrow subset of indications in drug products. So we tried to build that in. How do we do that? Well, we do it with a configurable modular platform, that allows us to drive faster optimization and have modules that suit the specific program needs. And that optimization is built off of a toolkit. with, that's driven with QBD guidelines that, allows us to quickly maximize efficiency, maximize productivity, and minimize, um, things like, contaminant profiles, minimize, and minimize losses across steps. And then the whole thing has to be data driven, right? We want to leverage that long history of data and know-how, but at the same time, we want to also leverage contemporary data that's specific to the program, so that we can, you know, build the highest yielding, highest quality product with the most predictable performance. So, this is the philosophy that we use when we developed our AAV curator, and this is the philosophy we wanted to employ when it came to LVV curator. So, the curator platform, as it looks for AAV, you know, is again modular. You see different upstream applications and modalities for manufacturing with suspension and adherent options to optimize transfection, media release, DNA ratios, enhancers, and then um, options to adjust for scale and CQA needs on the downstream as well, whether that's an ultra centrifugation application or an affinity or an ion exchange uh, column to do empty full enrichment, all the way through final formulation, different approaches to that, so that we can be flexible to scale, and then different vial formats and fill volumes, to suit those needs of an ocular indication that's for an ultra rare disease that has super low yield requirement but very specific CQAs, to a systemic application that's for a highly penetrant disease that affects adults that's going to require massive scales. It's hard to build a one size fits all platform that can do those things. That's why we take this modular approach. So, the it all starts with analytics. Um, we can't begin to develop something unless we can understand it. So, we have very deep expertise in analytical development and QC testing. It spans that entire timeline, just like the AB manufacturing does. We have a long list of in-house assays for raw material testing, all the way through in-process release and stability. Um, it really gives us end-to-end capability, and we wanted to leverage that for lentivirus as well. But we had to start somewhere, so we wanted to start with those enabling assays, and those namely are the titering assays, because that's what um, you know assessing titer is how you really drive quality from the onset. If you're developing a high yielding process, it tends to the quality attributes tend to follow suit. We have to pay very close attention to them, but these are the first things we want to look at. So we didn't just want to look at um, one titering assay alone with lentivirus, because lentivirus being enveloped and far more susceptible to stability issues and processing. We wanted to look at the relationship between physical titer with our P24, genomic titer with RNA extraction and an RT-qPCR, and our transduction assay, which this is the bread and butter assay, is functional titer for lentivirus. So we wanted to see how all three work in relationship. So moving on to upstream production. So again, we wanted to see a systematic approach to our upstream development, very stepwise, optimizing at each step from, the cell line, the media, reagents, all the way through our timing of transfection and harvest. Um, don't have time to get into, like I said, I could talk to you on each one of these things for 20 minutes. So I'm trying to pick the highlights. So we'll start with cell line selection. We have a really great cell line in our Curator platform already. So we were really keen to see how well it performed with lentiviruses alongside AV. And we were very pleasantly surprised to see that our current Curator cell line performed very well. We compared it to commercial, readily available, popular commercial cell line, and in our hands, at least, it performs as well or better. You can see here in the suspension, slightly better, and in the adherent, it's comparable. Um, and then moving on to growth medium selection, we looked at multiple parameters with growth media, from growth kinetics to cell health, and um, but ultimately looking at overall yield performance for each one. Um, we saw a myriad of results from very poor performance to excellent performance. So but at the end of the day, it makes selection relatively straightforward. You also notice that the yields are going to vary a little bit here. I should qualify this, um, because we're looking at we wanted to, rather than normalize data across all the experiments, we found it much easier to normalize data within the experiment based on yield. So you're seeing different yields because it's different volumes, but those volumes based on scales that we worked at, whether it was a flask or a hyperstack, um, are all normalized within the experiment, just not across experiment. So if you're looking at this with a keen eye, you might notice that. That's why. So, um, so yeah. Then moving on to transfection reagents. Very clear here. We tried the virus gin versus some other reagents, and you can see this is a comparison of two. Um, you know, clear winner, made that easy. So the transfection reagents, obviously a critical component. So and then another, what can be a critical component we found is our supplement and enhancer selections. It's enhancers incur additional cost, as do supplements. Supplements far less so, but um, the differences can at times be dramatic. So they're well worth using in a production platform. But you want to move the needle significantly by using them. You don't want to see a 10% increase. You want to see fold increases, which looking here across multiple different supplements and our suspension platform, we're seeing at least twofold or better increases with the supplement, and same thing with the adherent. But it isn't just the supplement itself or the enhancer, but it's also the timing of addition. So we wanted to look at that too. Um, we saw marginal increases based on timing, but massive increases in the adherent, and then in the suspension, I should say, the massive increases in adherent. Um, you know, in this case, logs. So um, again, can make a huge difference in overall yield performance of your platform with the use of enhancers. So we have a high yielding production, upstream production platform. Now we have to purify it. This is where it gets tricky with lentiviruses. Again, envelope virus, much more susceptible to stability issues, to shear force, just in processing issues in the downstream. So we needed to take particular care here and really look at recovery rates. So we looked at clarification filter studies, tangential flow filtration, again, stepwise, right? Through chromatography, and then eventually, final formulation. So, beginning with clarification, we looked at multiple different filters. We're looking for high recovery. Um, we typically see an AV better than 85%. So that's sort of, we were looking for that same bar here, which we were able to achieve. You can see filter 3 is up in the 90% range. But then we also wanted to look at the performance of that filter across different conditions, in this case, flow rates. Um, so we're looking here at pressure change over time expressed as volume, and we're seeing really nice curves, pressure curves as the as we flow media over the filter. So I would say a high and a higher flow rate, or low and high flow rates. You're seeing very comparable curves. That's what you're looking for to show that you have a robust process in your filtration. And then in tangential flow filtration, here's where we have to pay really close attention to shear and membrane fouling. So, again, recoveries, we want to be in the 85 plus range. And then we also want to look at or transmembranic pressure, TMP, as it's expressed as, in terms of flux over time or flow over the membrane over time. And what we want to see is nice flat TMPs as we increase flow rates and as that flux increases. So, we were able to achieve really good flat TMPs at good flow rates. You can see, where it's nice and flat, and then you start to see the slope as pressures increase over time. So, both cases here, high recoveries, good flow rates, good transmembranic pressures in high flux. This is our pre-chromatography TFF that we looked at here. Same principles apply in final formulation. It's a much more purified state. So, we need to pay particular attention to recovery here. So because it's more purified it is, the more susceptible it is to shear. But we got, good solid recoveries for both membranes. So, in this case, we're looking at multiple membranes that both have performed quite well, which is, fortunate for supply chain reasons. We have flexibility there. And then moving on to ion exchange chromatography. So this is the workhorse purification step in our LBB platform. We looked at multiple different modules, which consisted of different different resin chemistries and buffer matrices. What we were looking for is an elution profile that had very high fraction percentage recoveries in a single fraction. Now that's indicative of a good sharp single peak. So, module 1, 2, and 3, you can see you have recovery percentages spread across multiple different fractions. But in module 4, the vast majority of the viral vectors are all in fraction 1. So, and then you just look at the chromatograph of that module from the wash and elution step, and you see, a really nice, clean, sharp peak with no shoulders, which is, which I would consider, an ideal circumstance. So we've looked at each individual step, right, from the upstream to the downstream, and now we have to put them together in the aggregate and see what the overall performance is from the time we harvest all the way through. What we're looking for is a good mass balance and recoveries, getting what we expect. And this is where we're really looking at that relationship between, our functional titer of transducing unit as opposed to in conjunction with rather our viral genome and viral protein physical titers. So you can see, very nice stepwise recoveries, good mass balance. Are at sterile filtration, this would be the drug substance stage, recovery of greater than 15%. We're like 16.5% here. And the relationship is close to 1-to-1. And this is at a small scale. So increasing scale, going from, and we're looking at adherent here, increasing scale from a single cell 10 stack to a 5 HYPERStacks, which is 20 liters of feed stream. We want to see the same sort of profile. And we do. we're starting to see some assay variability here, but you still mass balance is very on par. Recoveries are even better. And I do have this asterisk here, this chromatography step, because we did run into some challenges with the load at this scale, which we've worked out. But why would I point out a flaw? Well, it's because it demonstrates the robustness of the platform. Even though we had issues with the load and chromatography, we still got an exceptionally good recovery at the end of the day of greater than 25%. And those relationships are fairly flat one-to-one ratios for the most part across the board. And then, looking at scalability, not just the 20-liter process alone, but the aggregate across multiple scales. You want to see, if nothing else, that you're not losing productivity, but in this case, we're actually gaining productivity, which is excellent, excellent news. We couldn't have asked for better. So, going from a single cell stack to 2 cell stacks, these are 10 stacks, to a HYPERStack train of 5 HYPERStacks manifolded together, we're seeing increased productivity as we move up in scale, which couldn't make me happier. So, at the end of the day, you're left with a platform that looks a lot like that AV platform by design. It has, uh, you know, flexibility in upstream modalities to address scale, either suspension or adherent. It gives us flexibility in our plasmid and optimization capabilities with our plasmid ratio and enhancers. Um, Flexibility in the downstream with our filtration steps and our chromatography steps, and then ultimately, you know, the same final fill flexibility with volume and vial format. Um, Again, leveraging all the good things that a platform brings to the table with platform analytics, standardized costs of goods, uh, you know, scale down models for performance qualification, all of those things, but a modular design that addresses uh, those issues that come with uh, different indications, whether it's an autologous, a single dose, or a large allogeneic application. It can handle all of that and everything in between. So, with that, I want to thank you for listening to me, and provide some acknowledgments to Tiffany, Michael, and Samir, and that all three are rock stars in our PD department and did all the heavy lifting with all of this platform development. So, thank you very much.